All NEET aspirants, you are listening to podcast Botany for NEET by S. Ramakrishna. In this podcast, biotechnology, very important topic, a tricky conceptual topic. Keep the NCERT textbook open with you, if possible, pen and paper. The delivery unravel all the knots between the lines, and finally you are going to feel easiness, and at the time of examination you can recall the logic between the lines delivered to you, because it is not possible to recall the sentences you underline at the time of examination under pressure. So don't skip, don't scroll, listen with attention. In biotechnology, before knowing much about the biotechnology present in case of the text, first you have to know about why it emerged, how it emerged. Right from the beginning, humans started observing the nature. Why humans started studying the nature? Curiosity, or for comfort, or for survival? Initially, curiosity. Humans first observed the nature, understood the nature, then finally utilized the nature for his own benefits. So, technological applications to living organisms to derive products useful to the humans is nothing but biotechnology. Living organisms from technology, products, biotechnology. So, biotechnology born out of curiosity but shaped by human needs for comforts. So, ever since the period of Rene Descartes, who was naturalist, philosopher, mathematician, biologist, from his time period onwards, all our human knowledge with regard to the natural sciences directed to develop technology. Technology again directed to develop products for human comforts, creature comforts, and add values to the human life. Whole approach of understanding natural phenomena, it became anthropocentric, who observed the nature and natural phenomena, but latter who utilized it for his own purpose. So the view was anthropocentric because major utility of the biological world viewed by the humans as the food source. That's why we are calling it as anthropocentric view. The physics and chemistry gave rise to technology, industry, engineering, again which directed towards human comforts and human welfare only. And offshoot of the modern biology, with the advent of molecular biology, the biotechnology emerged. So it is offshoot of modern biology. The biotechnological products brought great qualitative improvement in case of the human life, lifestyle change, expectancy of the life period increased with the discovery of the new medicines and food products. So lifestyle changed because of biotechnological products. And in this topic, the biotechnological principles and process, and at last, what are applications of biotechnology? Next, Herbert Boyer was born in western Pennsylvania, where railroads and mining are the main destiny, that is the employment for youth, who graduated from Pittsburgh University and postgraduated at Yale and worked as assistant professorship in University of California at San Francisco, who studied couple of restriction enzymes from E. coli with useful properties. That is, it cleaved the DNA in a particular fashion, results in two strands, that is, the clipped ends having single stranded at the end of the double strand, that is overhanging ends, single stranded ends at the end of double strand, and the clipped ends readily pasted as a precise exercise, exactly pasted the two fragments, clipped ends readily pasted, we call it as DNA splicing. And Herbert Boyer, who met Stanley Cohen at Hawaii, a rich and rewarding conversation between the two scientists. By the time the Stanley Cohen, who had been observing ringlets of DNA, circular, naked, double-stranded DNA, which is extra genomic, floats in cytoplasm of certain bacteria, what we call plasmid. The plasmid replicate independently of coding strand. That means it replicate independently of main genome. Cohen conducted an experiment, simple experiment, who isolated the plasmid and reinserted into other cells. So, isolation and transfer technology developed by Cohen. DNA splicing technology by Herbert Boyer. So, combining the process of both Herbert Boyer DNA splicing technology and Stanley Cohen, that is isolation and inserting technology, combining the process, the desired DNA configurations introduced into the bacterial cells that act as a manufacturing plant for specific protein. Altered plasmid, altered DNA introduced into the bacterial cell by Stanley Cohen and Herbert Boyer that act as a manufacturing plant for specific protein. Great breakthrough in biotechnology. How it is a great breakthrough in biotechnology? Cut, paste, insert. The whole biotechnology is based on that principle. So in next episode, the biotechnology principles and the process. In this, you go through a few questions. René Descartes belongs to 17th century. Biotechnology is offshoot of modern biology. Our view towards the nature always it is anthropocentric. That's why biology registered very limited progress. Next, Herbert Boyer developed DNA splicing technology. Cohen, who developed isolation and reinsertion of the plasmid into the cells. Unaltered plasmid transferred into the cells by Cohen. Altered plasmid transferred into the cells by Herbert Boyer and Stanley Cohen. The clipped ends readily pasted as a precise exercise by Herbert Boyer.